my absolute pleasure to introduce Hongbo Chi from St. Jude Children's Research Hospital in Memphis. I thought I was slick by taking over the seminar series at LJI so that I could invite scientists that I admire without running it by anybody else. But once word got out Hongbo was coming, the whole Mesa kind of got involved. So it became a collaborative seminar this time. So we're really excited to have him. I'm going to keep it short, but some of the honors that Hongbo's had just in the last couple of years is he's been awarded the Outstanding Research Award. He's got an NIAID Merit Award. He's been referred to as one of the most highly cited scientists in 2020 and 2021. And so I looked up his H-index, which is 53, on 2 different websites. And for relative, I'm not even listed on the website. So you can tell this is going to be a fantastic talk, and I'm really looking forward to it. So with no further ado, Hongbo.
Well, thank you so much, Sam, for the kind introduction. I'm very honored also to be invited by both Sam and Michael. And actually I have to brag about it. Angela invited me a couple of years ago for the LGI talk. I gave it virtually. So I'm so excited to be here in person and see Ananda's face. The long-standing interest of my group is to study immunometabolism, especially how the immune signaling crosstalk with metabolic program and how they regulate immune responses and in particular T-cell mediated diseases. That's why for today's discussion, I'd like to be focusing on work in T-cell metabolism and also the implication in immuno-oncology. To study the interplay between immune signaling and metabolism, we are interested in several kinase pathways, especially mTOR. And we also explore how this kinase pathway crosstalk with the cell metabolic programs, including bioenergetics, such as glycolysis and oxidative phosphorylation, cell biosynthetic activity, and more recently we are exploring how nutrient and the metabolite can serve as signaling role to regulate immune responses. So therefore, given this extensive crosstalk between signaling and the metabolism, we consider this as a two-way street or bidirectional metabolic signaling. And within that underlying theme, we explore both T-cell metabolism as well as how T-cell immunity is cross-regulated by metabolism of antigen-presenting cells, dendritic cells. And for today's discussion, I'll be entirely focusing our work on T-cell metabolism. And first, I'd like to give some introduction to our work in actually integrating system immunology with T-cell metabolism. And then I'll be focusing on work in Treg biology. So it's going to be 2 topics. Metabolism, as we know, is a fundamental requirement of all living cells. But instead of simply serving as housekeeping function or this essential role of cell growth, the metabolic activity is a very active and dynamically regulated process. For instance, for highly proliferating cells such as cancer cells, they will upregulate glycolysis even in the presence of abundant oxygen. This is known as aerobic glycolysis or the Weber effect. For activated T cell, partly because their massive rate of proliferation, so they will upregulate glycolysis, not a surprise. However, what was surprising was a collaboration with Doug Green, actually a few years ago, when we found that the cell glycolytic activity, the T cell glycolytic activity is much highly upregulated when T cells develop into effector cells, including Th1, Th2, Th17 cells, versus their differentiation into induced Treg cells. And we further show this stronger glycolytic activity for effector cells, especially Th17 cells, is important to support this cell fate differentiation process. Over the past few years, many metabolic programs have been identified in T cells. As a matter of fact, T cells, each step of the development or activation of function requires active reprogramming of their metabolism, starting from thymic development to peripheral activation and then differentiation into effector, regulatory, or memory cells. Moreover, recent study highlights the importance of metabolic reprogramming for T-cell mediated diseases, including infection, actually including the COVID-19 infection, the immune response towards COVID-19 infection, actually SARS-CoV-2, as well as many autoimmune diseases. These are mediated by the unique metabolic programs, the nutrient transporters, and also the driver genes that's associated with the T-cell activation and the differentiation state. In the case of highly immunosuppressive tumor microenvironment, it is often metabolic challenging. For instance, T-cell function and metabolism are often limited by the nutrient signals available to them, including the mechanism of nutrient partitioning. This is how the nutrients are distributed among different cells in the tumor microenvironment, nutrient deprivation, and also build up the toxic nutrient and metabolic products. And the last panel showed the regulation of the inhibitory pathway inside the cells. So therefore, for effective T-cell therapy, we need to restore and promote the metabolic fitness of T-cells. Given this consideration, then what are the important players of the metabolic pathway in T-cells? So in this recent review in Nature Review of Immunology, we just highlight several of the well-studied metabolic pathway in T-cells, including glycolysis, glutamine metabolism, mitochondrial metabolism, and also the lipid metabolism. And we describe how this metabolic program can be shaped by the immunology signal, the T-cell receptor, co-stimulation, and cytokine signaling, the signals 1, 2, and 3. At the same time, we also start to appreciate the importance of the nutrient in the microenvironment, such as glucose, amino acid, or minerals, and how this nutrient can impact the T-cell metabolic program and also signal transduction pathway is really emerging area of interest. So therefore, we like to call them collectively as signal 4 to regulate T-cell immunity. And among the central regulators of many of these metabolic processes is a kinase known as mTOR. mTOR serves as the central regulator of cell metabolism and cell growth, and which functions through one of the two kinase complexes, mTOR1 and mTOR2. A unique feature of mTOR signaling is its ability to integrate signal from either outside the cell, such as immunology signals, growth factor, and nutrient, or many times it reflects the cell intracellular metabolic state. And we are very interested in how mTOR and other metabolic pathway can regulate T-cell responses. And from this perspective, we study both the conventional T-cell activation and differentiation, and as well as the Treg biology. What we'd like to draw this simple metabolic diagram with these simple cartoons or many of these linear arrows. We know in reality, the intracellular metabolic network is incredibly complicated process. For instance, the metabolome is much more complex than the protein or transcriptome. It's more difficult to measure in general, and also it's very dynamic as it frequently involves metabolic feedback, feed-forward, adaptation, and crosstalk processes. Partly because of this huge complexity, we believe a system-level understanding and system-level investigation is critical to study immunometabolism and also immune-mediated diseases. And system immunology refers to the use of omics and large-scale data set to understand immune system function and the underlying mechanism. Of course, it involves deep profiling at a different level, at the population level or single cell level. But more importantly, it is the data integration that's crucial to gain biological insight and disease target. So from that perspective, let me just share with you some of the system tools we use in our toolbox. This includes the hidden driver analysis, which is in collaboration with Yang Yu at Jude. So the idea is many of the important driver genes, they may not be regulated at mRNA or protein expression level. However, if they're important, their protein activity should be reflected by the target gene that are regulated by them. So therefore, by profiling the target gene expression, then we use a computational program to predict the driver molecule. Therefore, we call them hidden driver analysis. Also, in collaboration with Jude and Jude, we extensively apply proteomics profiling. So this goes with total protein, phosphoprotein, as well as the protein-protein interaction or PPI network analysis. And also, more recently, we actually heavily apply in vivo CRISPR screening as a way to identify the new driver and the new target in immunometabolism. And in the next few slides, let me just share with you give you some examples of these 3 different system tools. The first one, as I mentioned, is hidden driver analysis. And here, in order to identify this hidden driver, basically based on the protein activity, we apply the transcriptome, whole protein, and phosphoprotein profiling. And then we also reverse engineering this network that reflect the driver and also target gene expression. So therefore, by profiling those target gene expression, and also integration with this network, now we're able to predict the driver molecule that's important for immune responses, also for immunometabolism. And more recently, we applied this network approach to single-cell data sets, including single-cell RNA sequencing and single-cell ATAC-seq, which help us to build intracellular drivers as well as the intercellular, the cell-cell interaction driver molecules. The second system approach we apply is system proteomics. This is in collaboration with the Jude group. We did this one of the first proteomic profiling of naive T cells activated at a different time point of following T cell receptor stimulation. So altogether, we're able to capture almost like 10,000 proteins and also over 10,000 phosphoproteins. So in a way, this is also equivalent to doing like 10,000 Western blot in one shot. And having obtained such data, then we also developed a network approach to integrate this data, try to get a sense of the biological process underlying, which allow us to identify the reprogramming of mitochondria as important to support T cell exit from the quiescent state. So therefore, at the beginning, I mentioned T cell will upregulate glycolysis upon antigen stimulation, but here we also show the mitochondria metabolism is also critical to support that process. So partly building upon this proteomic profiling, we propose the 6 hallmarks to drive T cell exit from quiescence. This is this include cell growth, cell cycle entry, cytokine signaling, anabolic metabolism, nutrient uptake, and also reprogramming of mitochondrial function. And we also highlight that mTOR and CMYC serve as a central hub to coordinate these signals, this intracellular metabolic signal with the immune and the metabolic cues the T cell will receive. Moreover, we can apply this metabolic quiescence and metabolic activation principle to different immunological contexts, including Th17 cell. We show that Th17 cells are metabolic heterogeneous. They include like a TCL1 positive, like stem-like subset or homeostatic subset, and a more terminal differentiated type 1 T cell-like mature cells. This is more pathogenic. So therefore, those 2 subsets can be defined by their unique metabolic programs. That's why we call them metabolic heterogeneity for Th17 responses. The third system approach we use, which actually became our favorite, is in vivo CRISPR screening. So let me just use one of our first example to show the ideas behind. Here we are interested in how the metabolic drivers can modulate T cell immunity. So in a way, we are searching for the negative regulatory molecule that when target in T cell will drive or boost T cell immunity towards tumor. So therefore, we build this metabolic library that target over 3,000 known or putative metabolic regulators. Then we'll transduce this library into tumor-specific T cell, and followed by parking those T cells onto tumor-bearing mice. And 1 week later, we ask those T cells to compete, then trying to find the winners. And this allows us to identify Recognize-1 as the very top hit. That's in the absence of Recognize-1, there's massive accumulation of T cell in the tumor microenvironment. And this also shows much better anti-tumor immunity. I appreciate actually Ananda and Miguel's news and view to highlight the importance of Recognize-1 and also the program regulated by Recognize-1 to affect anti-tumor immunity. But having identified Recognize-1 at the top inhibitory pathway for anti-tumor immunity, then we look for the target gene by which Recognize-1 can function. And Recognize-1 itself is an enzyme that will degrade mRNA. There's thousands of targets. So therefore, it's very hard to identify the right target. So here, again, the CRISPR screening is highly powerful in that we did this genetic in vivo rescue CRISPR screening by co-transducing T cells with Recognize-1 guide RNA together with the genome-wide library. And we look for the guide RNA that can rescue the Recognize-1 knockout cell phenotype. This allows us to identify BATF as a crucial target that function in the to drive the Recognize-1 knockout phenotype in terms of the boosting T cell killing activity. At the same time, we also identify TCF1 as another key target affected by Recognize-1. So therefore, in the absence of Recognize-1, both BATF and the TCF1 expression gets upregulated, then we can boost both T cell killing activity as well their long-term persistence. This is part of the reason we believe that can explain why the Recognize-1 knockout T cells are highly effective in killing tumor. So more recently, we're trying to move this into, hopefully, into early phase clinical trial. In collaboration with many St. Jude investigators, we developed this GMP-grade guide RNA targeting Recognize-1 in human T cells, and then in human CAR T cells as a way to treat B-ALL, the leukemia that affects a large number of pediatric patients. So in this proof of principle study, we transfer the tumor cell, the human tumor cell, into the ASC mouse model, then followed by the transfer of either Recognize-1 wild type or Recognize-1 knockout CAR T cells. As you can see, compared with the Recognize-1 wild type CAR T cells, those lacking Recognize-1, the Recognize-1 knockout CAR T shows much better therapeutic effect. And overall, we're quite excited about this project as it shows an example of truly unbiased discovery of a novel target to shape cancer immunotherapy, and also is this project establish a clear path to clinical translation.
What do the survival curves look like?
I skip it, but it's quite good. It's not 100%, but it's quite I think quite good. I mean, at least in the molecule we knock out, that's the best one we got so far. Now for the rest, like 15 minutes, let me share with you some of the unpublished study. Now we apply the similar actually CRISPR screening, and we screen for epigenetic factor. Part of the reason is the epigenetic pathway will constantly crosstalk with the metabolic pathway. And part of the reason actually, this is a really nice collaboration with Doug Green, who actually, I'm sure everyone here knows Doug's name, who used to be actually at LJI. So here we use the CRISPR screening, try to find the epigenetic factor that in its knockout will boost T cell memory. Here we use actually Listeria over infection, very simple infection model, partly because we can analyze T cell responses like after 1 month of Listeria infection. We can isolate different T factor terminal effector cells or memory precursors, but also we can compare the T cell enrichment at the later stage versus the early stage of immune responses. From this epigenetic library, we identify the SWI/SNF complex, the conventional BAF complex within the SWI/SNF big complexes. It's crucial inhibitory pathway for T cell memory. And in the absence of those genes, they all belong to the CBAF complex. You can see this stronger T cell memory responses at the expense of effector responses. So this is a comparison of memory phase and also the effector phases of T cell responses. The BAF complex is part of the SWI/SNF complexes. And also the SWI/SNF complex itself has three different subcomplexes, including the conventional BAF complex, the PBAF, and the NC-BAF complex. And overall, these are the important chromatin remodeling complex that can affect gene expression in different types of cells. So having identified the CBAF complex as an important regulator of CD8 T cell memory, then we did the validation experiment. Here, again, this is quite a routine Listeria overinfection. Then we examine the T cell accumulation in different lymph organs. We found that indeed, as the data shown from the CRISPR screening, when we knock out either the SMARCD2 or RAD1A, each of them belong to the CBAF complex, there's more memory T cell generated in different organs examined. And also for this, upon the re-challenge of the Listeria infected mice, we found that those T cells, those knockout T cells shows better recall response, suggesting that there's better memory formed in the absence of those CBAF complex, but also there's better memory function in the absence of the CBAF complex. As for the molecular insight, we noticed that the CMYC which happened to be included in our library, also turn out to be an important regulator of T cell memory. And Doug Green actually have this really nice insightful paper suggesting how the CMYC, that's their expression, is very important for T cell to make their decision between effector versus the memory cells. In that the cells with higher CMYC expression are prone to become effector cells, while those with actually less MYC expression turn out to be more likely to become memory cells. So therefore, we explore the interplay between the CBAF complex and this transcription factor CMYC. And indeed, as I mentioned, the CMYC high cell will contain higher CBAF complex expression, while the CMYC low cell, which turn out to be, those are the cells that are more prone to become memory cells, have less BAF complex expression, suggesting that there's potential interplay between these two pathways, the CMYC and the CBAF. So we went deeper. Actually, many of the studies I have to say is done by Doug Green, all in Doug Green's lab. So what I did here is to further examine whether there's actually physical interaction between these two factors. This is the common immunoprecipitation Western blot analysis. And indeed, when we all found out when he immunoprecipitated a RAD1A, he can put down CMYC. And conversely, when he immunoprecipitated CMYC, he can put down a RAD1A, suggesting that the physical interaction between these two pathways. Moreover, from the transcription factor binding size, there's extensive degree of overlapping binding. In this cut-around analysis, is a low input chromatin binding assay, and the CMYC binding site and RAD1A or BRG1 binding site shows a great level of overlap. And many of these overlapped binding sites include the genes involved in CMYC, of course, but also the metabolic pathway, including mTORC1, T cell differentiation, and also T cell activation. So therefore, CBAF and CMYC can physically interact with each other, and they appear to act in concert to establish the chromatin state. To further explore the functional effect of this interaction, then we made knockout cells, then examine the other factor binding. So basically here we've made the MYC knockout cells, then examine the CBAF binding. And indeed we found that with the deficiency of c-MYC, the binding of this CBAF is diminished. This is just a control experiment. Of course, with c-MYC knockout, c-MYC binding is greatly reduced. The reciprocal experiment is to examine the binding of c-MYC to the CBAF deficient knockout, to the CBAF deficient cells. So for instance, in the ARAD1A knockout, the c-MYC binding to selective target, including granzyme B and the TBAC21 is reduced. So therefore, this data also reveal not only these two factors can interact with each other, there's also reciprocal dependency of them to bind to their respective binding site. Further revealing these two pathways can act in coordination to regulate T cell fate. Finally, we examine the physiological impact of deleting the CBAF complex for T cell mediated and anti-tumor immunity. So here's one, survival curve. And in the absence of SMARCD2, the tumors, when we transfer T cell with the deficiency of SMARCD2, those T cells can control tumor growth better, and the tumor size is reduced, and the survival is modestly improved, I would say. But perhaps the more remarkable finding is the drug inhibitor study. In this CAR-T model, we applied the CBAF inhibitor during that CAR-T culture period. Then we transfer those, we wash off the inhibitor and transfer them onto this solid tumor berry mice. So therefore, this is only very transient treatment of this CAR-T cell during the in vitro culture. And what we found is those T cells, even with this very transient inhibition of CBAF complex, they show much better persistence in vivo. The T cells express the luciferase, which can be directly monitored in vivo. And consequently, the T cells, the CAR-T cells, even with very brief treatment of inhibition of CBAF complex, they show better anti-tumor immunity. So therefore, transient inhibition of CBAF can promote CAR-T cell persistence and anti-tumor function. To summarize, from the unbiased in vivo CRISPR screening of epigenetic factor, we identify CBAF as a critical inhibitor of CD memory. We also found that mechanistically, CBAF complex and c-MYC, they can interact with each other and also regulate their the reciprocal target for those two pathways. And also, the CBAF appear to be an important target that can be reprogrammed for better anti-tumor immunity. Yes?
Done ATAC-seq to figure out which accessible regions are changed?
Yeah, we did that. I did not show the data here. There's quite a number of allele, binding chromatin region are affected by actually either of them.
Could you relate them to the genes that change expression?
I'm sorry?
Could you relate them to the genes that change the
Yes,
expression?
yes, right. Many of them we do the, actually sometimes use another program, right, to coordinate the ATAC-seq data with RNA-seq data. Many of them are shared, so.
Have you done the CBAF inhibition study for the memory response?
No, we have not. We have not.
Like, what would you expect if you just
Right.
do a transition inhibition? Would that affect the long-term memory formation?
Right. What we did, I can explain a little bit more detail what we did, time point. We found actually the first 48 hours is crucial for the inhibition to work. If we wait 48 hours, then start drug treatment, in the CAR-T experiment, it's no longer have the effect. So somehow in that magic window, that's where the CBAF inhibition will be important. Doug's interpretation, he likes the asymmetric cell division, but whatever which model, that model may or may not be, be totally accepted, but I think that time window is certainly crucial. Also, this is also the window, that's what we call them exit from quiescence. because that's really the window with massive protein, epigenetic reprogramming. So we still don't, yeah.
Are you blocking activation? Are you
Actually,
suppressing
if activation gets a little bit reduced, but not much. That's why,
by what criteria?
I'm sorry?
What are your criteria for activation not being reduced much?
Cell number, right? We look, because we do the short treatment in vitro, right? We transfer them in vivo. We look for the short term, like after a few days later, looking for T cell number. They reduce, but very modestly.
And what about cytokine expression?
Cytokine expression, actually, we also examine interferon, which interferon is not affected.
Yeah,
So,
I'm
I mean,
just curious to see curious whether you think that some other aspects of activation are suppressed to give you increased memory function. You would expect.
Right, we would expect the activation would be reduced, which we see. I mean, as I said, like cell number reduced a little bit, but later on they'll be met actually more cells later on. Right. Okay, all right, thank you. So overall, the idea to build upon this screening data, also the target we identify in CRISPR screening, build better cars, and better cars that can last. So what have we learned from this part of the discussion, from the in vivo CRISPR screening? We believe this is one of the best way to discover new biology, new driver genes, and also new target. And also, I did not have time to show here the extensive discuss it, but this is very crucial way to identify new mechanism, such as the genome-wide rescue experiment, rescue screen we did to identify the target for Regnus-1. And using similar screening platform, we also explore the CD T cell differentiation in different contexts, including identification of the fucose, which is a unique sugar that's important to link to CD T cell fate decision. That's because it's a way to regulate NASH signaling. And we also identify the phospholipid known as phosphatidylethanolamine is important to drive follicular helper T cell differentiation, in part by stabilizing CSCR5. So having tasted or experienced such a power of system immunology, so actually at St. Jude we have this blue sky project. This is really only the sky's the limit. Then we propose to use a system immunology approach, especially CRISPR screening and this computational approach, to identify new target for pediatric cancer, because St. Jude is a hospital for pediatric cancer. On one hand, we propose use in vivo CRISPR screening in cancer cells or T cell or CAR T cells for the pediatric cancer, including the leukemia, brain tumor, and also solid tumor. And on the other hand, we apply the hidden driver analysis and also combine that with single cell profiling, try to build the tumor microenvironment interaction maps, and by explore both the intracellular and intercellular driver genes. So this is where the first topic is about. Then I'd like to switch gears and move on to the second part of the discussion, talking about the nutrient signaling in Treg cell. So basically, this is the dark side of T cell immunity for tumor. Treg cells, as we know, are crucial to mediate immune homeostasis, but on the other hand, Treg are the major barrier, one of the major barriers for effective tumor immunity and tumor immunotherapy. So therefore, how to find the balance? Find a balance that can still maintain immune homeostasis, but will disrupt tumor-specific Treg function has been an important question in the field. Our interest in this area came from this, actually again, somewhat surprising finding. It's surprising because mTOR was generally considered as a negative regulator for Treg cell function. But what we found is when we knock out Raptor or mTOR1 function in Treg cells, the mice die from inflammation due to the profound loss of Treg cell function. Mechanistically, we propose mTOR1 is important to drive anabolic metabolism, especially the mevalonate pathway, and then this is important to drive Treg cell proliferation and activation. Having established mTOR1 as a critical positive determinant of Treg cell function, we next explore the upstream and downstream pathway by which mTOR1 can function. For the upstream pathway, we define the two amino acids, in particular the arginine and leucine, are crucial to activate mTOR. And we call this a licensing role because despite the critical importance of T cell receptor to activate mTOR1, it is unable to do so in the absence of amino acid. So therefore, we propose the amino acid-dependent mTOR1 activation is important to license T cell induced mTOR1 activation. As for the downstream pathway, I mentioned mTOR1 is important for movalent metabolism. But further downstream, it's not actually the cholesterol itself, but this isoprenoid appear to be very important in mediating the movalent metabolism in regulating T cell function. Those isoprenoid are lipid moiety that can be added onto the target gene, target protein such as the small G protein RAC. So through the post-translational modification process known as protein prenylation. This includes protein phenylation and also granulation. Basically, this is a unique PTM mode of regulation. And over the years, we made actually many FOXP3 dependent deletion of Raptor and many other mTOR and also metabolic pathway. And in each case, actually, each of this knockout we listed here, we can kill the mice by inducing the profound loss of Treg cell function. Well, these data are analyzed and show the importance of this metabolic pathway in programming Treg cell function. This pathway clearly should not be targeted for tumor therapy. So therefore, we have been also searching for the pathway that can mediate Treg function in a tumor-specific manner. And indeed, recently we found the de novo lipid synthesis appear to be one of those pathways that can function in tumor Treg specific manner. Song in the lab, talent fellow in the lab, she found that when she knocked out SCAP, which is an important adapter molecule for this group of transcription factor known as SIBP, when she knocked out SCAP in Treg cells, those mice are completely fine. They don't develop autoimmunity, but those mice can clear tumor in a much better way compared with wild-type mice. And similarly, when she knocked out fatty acid synthase, which mediate fatty acid, de novo fatty acid synthase, again, those mice with Treg specific knockout of FASN are completely normal, at least the way we look at it. No autoimmunity, but again, those mice will clear tumor in a much better way compared with wild-type mice. So therefore, we propose the lipid signaling in Treg cells is important for functional specialization of Treg cell in tumors. This is partly is to keep interferon gamma production in check. In other words, when we knock out SCAP, the interferon gamma gets excessively produced by Treg cells, which is detrimental to Treg function in the tumor. So altogether, we are quite interested in further exploring how lipid signaling and lipid metabolism can regulate T cell function, including the lipid synthesis, the movalent metabolism, as well as the lipid catabolic processes.
Hongbo?
Yes?
But when you knock out SCAP, you also inhibit SRBP2.
Yeah. So this is kind of equivalent to double knockout for SRBP1 and SRBP2.
Yeah, so, but on your slide, you say it's mediated through DNL, not through the mevalonate path.
Right, actually both. There are two branches.
Okay.
One is regulating the fatty acid synthase. The other one is movalent metabolism. Actually, both lipid-related pathways are
both fatty acids and cholesterol metabolites.
Exactly. Mm-hmm. Yes, absolutely. Yeah, yeah. We haven't been able to actually distinguish SRBP1 versus 2 function.
Okay.
And also, they actually downstream they go through different mechanisms, but both seem to be important for Treg function in the tumor context. So now we can add the lipid signaling, including SCAP and SRPP, or fatty acid synthase on the right side of the equation. So those, because they appear to be tumor Treg specific regulator, but then those knockout will not affect the systemic immune homeostasis. Having applied this reverse genetic or this condition knockout approach for the last 10 years, we got, especially in Treg cell, we got a little bit exhausted. That's why we try to use more system approach, try to reconstruct the entire signal network by which nutrient can signal. So that's when we started the CRISPR screening project by Ling Yun Long, a very talented fellow, trying to use the genome-wide screening in vitro, try to capture all the important regulator that can deliver signal from T-cell receptor and nutrient down to mTORC1 activation. And here the idea is he transduced the genome-wide library into the induced Treg cells, and followed by the stimulation of those Treg cells with T-cell receptor stimulation together with the nutrient. And here the nice part of it is for mTOR activation, there's very nice antibody for phosphorylation of S6. So therefore, we can sort S6 high versus S6 phospho S6 high and low cells, which would be a surrogate marker for mTORC1 high and mTORC1 low cells. This allows us to identify the molecule is important to either enhance or inhibit mTORC1 activity. Here, actually, indeed, we can rediscover many of the known regulators, including the mTOR raptor, all this molecule as an important positive regulator of mTORC1 in Treg cells, and also many of the known regulators for mTORC1. As a matter of fact, in total, we identified like 300 or 400 molecules that can be actually meet the cutoff, suggesting they're either positive or negative regulator of mTOR. Since this is a genome-wide library screening, then we also, to further validate this data, we did a secondary CRISPR screening by building upon secondary library. It's a smaller, much smaller library that only contains those positive hits from the primary screening. And in this secondary screening, we can again rediscover many of the known regulators of mTORC1. As a matter of fact, by comparing the first primary and secondary screening, 80% of hits are shared between the two screens, suggesting this is a very highly robust and reproducible screening platform. But we do have a problem, and that is we simply have too many hits. It's very hard to prioritize which molecule we should focus on. So therefore, this is our previous expertise by working with Jimin Peng group for the proteomics and also especially for the protein-protein interaction of PPR network became important. Here with the PPR network, now we can sort those different putative regulators into different beings, including the epigenetic regulator, transcriptional, or the translational machinery. And also we noticed there's also a complex known as gator 1 or gator 2. That's also important and be discovered in T cells. And gator 1 and gator 2 was the initial work from David Sabatini. They identified that gator 1 as a negative regulator of mTORC1, while gator 2 is positive regulator by inhibiting the inhibitors. We successfully capture both complexes in our T cell screening. In addition, we also identified SEC31A, which is molecule has not been implicated in mTOR regulation. We found that SEC31A co-segregate into the gator complex. So therefore, we focus on SEC31A as one of the important target of putative positive regulator of mTORC1. Here in this data validation experiment, we knock out SEC31A in Treg cells, induced Treg cells, then stimulate them with amino acid or glucose, as compared with wild type cells which will upregulate S6K or S6 phosphorylation. The SEC31A knockout cell has reduced ability to upregulate this mTORC1 signature phosphorylation events. Another event that that's induced by mTOR1 activation is mTOR translocation to the lysosome in an amino acid dependent manner. And we found that in T cell this is also true. With amino acid stimulation, mTOR will translocate into the lysosome surface, but in the absence of SAC31A, this translocation is nearly abolished. So suggesting that SAC31A is indeed required for nutrient-dependent mTOR1 activation. So what about molecular mechanism? As I alluded to, that SAC31 can co-segregate into the gator complex in our PPI network analysis. And indeed, we found these two proteins can indeed interact. In a way, SAC31A we found can interact, especially with SAC13. This is one of the unique component of gator2. When we immunoprecipitate SAC31A, we can pull down SAC13. The reciprocal experiment also true, although I did not show here. Suggesting there's interaction between the SAC31A, this new molecule we discovered, and SAC13. Moreover, with absence of SAC31A, we found SAC13 abundance is diminished in this Western blot analysis. But this diminished SAC13 expression can be blocked when we use MG132 to block ubiquitin-mediated proteasomal degradation. So therefore, SAC31A interact with SAC13 and promote SAC13 stability. In data not shown here, we also found that SAC13 indeed can involve the ubiquitination of this molecule. So this allows us to focus on SAC13 as an important mediator of gator2, and in a way that SAC13 itself is regulated by SAC31A. So that's the model and hypothesis we're testing. To test this, we first measure SAC13 protein degradation with cycloheximide treatment to block new protein synthesis, and you can see the SAC13 gets degraded. This actually measurement of the half-life of SAC13, and with SAC31A knockout, the half-life is shortened, suggesting indeed SAC31A can promote the protein stability of SAC13. SAC13 has a total of 16 lysine residues. We next explored how is SAC13 regulated by the proteasomal degradation and also polyubiquitination process. So in a way, we did selective immunogenesis to mutate each of the 16 lysine residue to arginine for SAC13. And we found that only when we mutate lysine 260, either alone or in combination with a neighboring lysine, we can remarkably boost SAC13 expression level. Moreover, when we make this mutant SAC13, we found the SAC31A is no longer functional. As you can tell here, with the mutation of lysine 260 of SAC13, the protein stability gets much better. But moreover, when we mutate SAC31A, we delete SAC31A, the SAC13 stability is not affected at all. So altogether, we conclude that the lysine 260 of SAC13 is important for its stability in a SAC31A dependent manner. Then finally, we went for the ubiquitin and also E3 ligase involved in degrading SAC13. Once again here, the PPI network and collaboration with immune pong is important, and we use the immunoprecipitation followed by mass spec analysis to look for SAC13 binding partner, which revealed actually SKP1 as one of the top hit. SKP1 is not E3 ligase itself, but it's a dimer molecule for a group of E3 ligase. And we found this from the this intacto analysis. We identified SKP1, and also in the individual validation experiment, we found those two proteins, SAC13 and SKP1, indeed can interact with each other, which can be controlled by SAC31A. And moreover, I'm just going to summarize data when we made double knockout of SAC31A and SKP1. Now we can rescue SAC31 knockout phenotype. In that, we can restore SAC13 expression. We can also restore mTOR1 activity. So therefore, this data reveal the SKP1 containing E3 ligase as an important mediator of SEC13 degradation. To summarize, from the integration of CRISPR screening and PPR network, we identify SEC31A as the part regulator of mTORC1, and mechanistically this involves the interaction with the GATOR2 component SEC13, and also the interplay with the SKP1 dependent proteasomal degradation system. So in the next couple minutes, let me just quickly summarize our data on the negative regulator side. Because SEC31A is a part regulator of mTORC1, but we're also interested in the negative regulator of mTORC1, which turned out, one of the negative regulators turned out to be the SAGA complex. We're interested in this process because we, several years ago, we proposed this Goldilocks principle for mTORC1 function in Treg cells, in that either too much mTORC1 activity or too little is detrimental to Treg function. So here we have the opportunity to identify a negative regulator of mTORC1, and then in those molecules' absence, then we can boost mTORC1 activity. We'd like to see the effect on the Treg function in vivo. So cut long story short, we identify CCTC11, which is the component for SAGA complex, which is actually another chromatin remodeling complex, as an important negative regulator of mTORC1. In this knockout, we found the mTORC1 activity gets elevated in response to either glucose or amino acid stimulation. And also mechanistically, this is associated with the excessive or aberrant expression of amino acid and the glucose transporters. In that, in the absence of CCTC11, the GLUT1 expression as well as several amino acid transporter gets upregulated, and when we did the double knockout, by knocking out GLUT1 in CCTC11 knockout cells, we can partly block this excessive mTORC1 activity. And also in vivo, to study the molecular, to study the in vivo function of CCTC11, in Treg cell, we generated FOXP3 specific deletion of CCTC11. We found that consistent with our in vitro data, mTORC1 activity gets up, FOXP3 gets destabilized, then with the Treg knockout CCTC11, effector T cell gets out of control, and the mice develop systemic autoimmunity, altogether highlighting the importance of the inhibition of mTORC1 by CCTC11. So altogether, by integrating the CRISPR screening with the PPR network, we identify a new driver for mTORC1 activity, that's SEC31 dependent protection of the GATOR2 complex from proteasomal degradation. We also identify the SAGA complex as an important regulator of nutrient transporter expression, including both glucose and amino acid transporters. In the now shown here, we also show the important actual, again, CBAF complex as an important regulator of nutrient sensors, such as Castor1. So altogether, we propose this three-tier regulation that can explain how the nutrient can be transported into T cells, how they're sensed by the sensors, and also how they can mediate signal transduction process. So this will conclude today's discussion on T cell metabolism, and also moving forward, we're quite excited to continue to apply the system approaches, including its functional genomics, the proteomics, the PPR network, and with the hope to identify new target and new driver molecule that's important for T cell mediated diseases. With that, I would like to acknowledge my group, including Sang and Ling Yun, for their contribution to the SCAP project and also the nutrient signaling project. Hong Ling, who worked with Doug Green's group on the CBAF complex. And also very grateful for many of my collaborators. Really found to work on, really to extend beyond traditional immunology study into the new space of system immunology. And also, really, I appreciate the support and collaboration with Ananda. So thank you so much for your time. Happy to answer any question you may have. Michael.
So, what's so special about Tregs that they depend on mTOR signaling and Th1s don't?
Th1 also rely on mTOR1, right? So I think mTOR1 is important for both Treg cells and conventional T cells. Treg cells likely require, at least under steady state, require a little bit more of mTOR1 because they are more activity in vivo. They have constant, for instance, the self-antigen recognition and the interleukin-2 signaling. So they have a higher metabolic state under steady state. So they have actually stronger mTOR1 compared with conventional cell, but actually they both require mTOR1.
So they have higher bioenergetic demands?
Treg cells?
Yeah.
That's really depend on which cell you compare Treg with. If you compare naive T cell with Treg, yes, they have higher metabolic state.
About activated cells versus Treg. They say
Right.
Tregs all the time monitored.
Right. Right. And if you compare activated T cell with Treg cell, this really is kind of a more or less equivalent, right? That's why the way I look at it, mTOR1 is not necessarily it's a lineage thing, but more about their activation state. Right? Lineage between Treg or non-Treg, but I think it's more about their activation state. Right? Mitch and Li Fan, you guys can compete. Sorry.
Another Treg question.
Uh-huh.
You reported that you didn't have spontaneous immunity, but the Tregs in the tumors weren't as effective. So what's special about the tumor environment? What subsets of Tregs?
Right.
Is it a hypoxic environment or something else?
Yeah.
What are your thoughts about
Okay, great, thank you. I'm waiting for that question to be to be asked. So we don't have a definitive answer, but we think the model is actually, which we favor the model is this involves the tumor microenvironment that is rich in lipid. Tumor microenvironment has like a nutrient deprivation with glucose or amino acid, glutamine included, but they're rich in lipid. So therefore, when we knock out SCAP in Treg cells, in the tumor, they can still uptake lipid, right? So in a way, the way we look at it, there's two pathways: de novo synthesis of lipid and uptake of lipid. We block de novo synthesis. Treg cells actually still alive. It's still actually they have the normal number in the tumor. We think that they do this by uptaking the lipid from the tumor, but still there's a detrimental effect is their function impaired. So therefore Treg number are still normal, which I did not show in the tumor, but their functional capacity is impaired. So that's how we can interpret all this data about the tumor specific contact.
Have you tried those SCAP knockouts in any liver inflammation or gut inflammation models, which might be lipid-rich environments?
We only tried the EAE model, which looked to be fine. But we haven't compared with other contact. Maybe Ananda can have more insight there. Hi, Li Fan.
Yeah. So I want to follow up Michael's question because your answer is that it's not more about the lineage, but more about the state of the cell.
Right.
But when you first do the screening, you're using this kind of in vitro Treg.
Right.
So they all have the T cell activation.
Right.
So when you do those screening, I assume you separate positive and negative, or you just use the total cell in the culture because not all of them are going to be positive. So I just don't know whether the thing that you identify here is restricted to positive cells, or is, as you said, if they're in the same state, then it's going to be the same.
Okay, great point, Li Fan. So technically, when we did the screening, we induced very high expression of FOXP3, like 95% pure. We didn't further resort them because in that case we have to do the FOXP3 reporter-mize. So we did not do that, but they're very, we're able to reach very high FOXP3 positivity. Also, we use the iTreg cells simply because these cells We can grow a lot of them. Also, they are very much better survival compared with in vitro Th1 cells. And also their dynamic range, I guess their induction of mTORC1 is very strong. There's several reasons why we choose iTreg cells, but many of those findings we can pull out what people publish in fibroblasts or cancer cell in T cell. That's why I don't think they're going to be lineage specific. It's more about their activation or all the nutrients present or absence.
So based on what you said, if you don't induce 95%, it's more like 50-50, then from that same culture, whether or not they express FOXP3 doesn't matter now.
Yeah, even we don't reach 95% pure. I just, of course, I'm a hand waving. We didn't do that in this department. I don't think it matters much. Right. Hey.
Hi, so it's a wonderful presentation. I learned a lot.
Oh, thank you.
Your presentation. So let me rephrase the question. So if you use an mTOR inhibitor, so you have these two effects on the Tregs, that you will reduce their activity, and at the same time, you will also be reducing the proliferation of the antigen responsive T cells.
Right, exactly.
But with the new TOR1 specific inhibitors, you can dial down very, very quickly. So do you expect it would be acting both would be immunosuppressive, or you would have a chance to low doses to be enhancing your response? That's
a great point. So in a way, the way I look at it, it's going to affect both cells, right? Conventional T cell and the Treg cells. It's going to be very highly dose dependent. If you give very high dose of rapamycin or mTORC1 inhibitor, you're going to shut down the immunity regardless of, because the conventional T cell are going to be all impaired. But if you give very low dose, which is actually shown by Raphael Matt, you can actually boost the T cell immunity, the memory cell generation. So first, it's very highly dose-dependent way, and then in that in vivo context, really, how the different cells contribute to the inhibition is hard to tell. So actually, in that case, we know for sure with that low dose of rapamycin, Treg are still perfectly functional.
Really?
Yeah. Actually, if anything, they get you have more Treg cells. So that's why sometimes also this is due to the partial inhibition of mTORC1 by rapamycin. It's only the S6K branches gets inhibited.
Right, but now you have the new Rapa links. They are very specific for TORC1.
Right. Actually, we have ongoing collaboration with Kivon, so we still explore that. Right, but thank you. Kivon Shukai, yeah. Right. Okay. All right, thank you so much for your time.